Welcome to the virus reproduction notes. This is lesson LS3B in the lymphatic system grid, and we're going to talk about specifically how do viruses reproduce, starting with what does it mean to have a viral infection. Now, we normally think of that just means that we're sick. We've got some symptoms like from the flu or chickenpox or the common cold, but what's actually happening? Well, at the microscopic level, what it means is that there are viruses using your cells to make more copies of themselves. This isn't written here on this presentation, but sometimes that means that your cells are being destroyed. Sometimes the viruses are releasing toxins. So there's a lot of reasons why your body has these things that we call symptoms. Your body is sending an alert to your immune system that something is wrong, there's something in here that's not supposed to be, and we need to get rid of it. Very briefly, the viral life cycle is just a bunch of steps in which a virus is first finds a host cell, reprograms it by giving its own DNA or RNA, and then letting the host cell make more viruses. In our last notes, we called that turning your cells into a virus factory. And we're going to talk about all the different ways that this new factory is going to release these products, sometimes in easy ways, sometimes in very destructive ways. I like this animation that I found that shows here's a virus entering a cell, more copies of the virus being made. Now they just sneak off and find a new cell. And this process just keeps repeating over and over. And of course, it makes more than four viruses each time. And it's just an exponential growth that keeps happening and happening until you have major symptoms or major problems from that virus, most likely. Now, the two cycles of viral reproduction are actually technically not seen as separate cycles. They're kind of just seen as two parts to a big cycle. It just the direction that the virus goes depends on what type of virus it is and what type of cell it's infecting. On your diagram, all you need to write on the left here is lytic cycle, Lytic, and then on the right it's lysogenic cycle. And you can look at this a little more closely later, but I'm going to explain how these two different pathways are different from each other. Here's the typical pattern that most viruses go through. It usually starts with some kind of attachment, and either the virus is just mounted onto the cell membrane and then injects its DNA in, or sometimes, as in this picture, the entire virus just comes right on in, and then we call that entry. It's either the DNA entering, or the RNA entering, or the whole virus itself entering inside of the cell. Then your cell starts making all the parts. It makes the genome, it makes the proteins to make the envelope, it makes the capsid, and then the next step is put them together. And all your organelles are working hard on these new instructions they got to make this thing they don't know anything about. The final step is release, and then they're off to infect a new cell. Now, this release can be different depending on what type of cell or what type of virus, and we're going to talk about the different ways that viruses are released. In the lytic cycle, it's considered the most common, the main method of viral replication. It usually results in the destruction of the cell. And it's named that way because in this final stage, when it's bursting, it's kind of like filling a water balloon with too much water. That process is called lysis, when a cell's membrane is destroyed because the cell just filled with too much water. That's not just water, virus, sorry. And the lysis is a noun. We can use it as a verb and say the cell is lysed, or the cell will lyse if it's filled with too many viruses. But then the word lytic is an adjective. So that's why lytic cycle is what we use when we're talking about lysis. I know it's words you've never used before, which is why I had this little section here on what does it mean and what form is it. So it means when the cell membrane is burst, and lytic itself is the adjective form of the word lysis. Excuse me. Now, there are also ways where the cell is not destroyed, where lysis doesn't happen. And there's two examples here, influenza and hepatitis B, two viruses that we'll talk about again in our next note. Influenza is one that comes right inside. The whole a whole virus goes inside the cell and is then makes copies of it, and then those copies leave gently. They kind of take a piece of the membrane with them, which they then use as a key to get into the next cell. And that's why they get recognized. That's why the immune system doesn't know what's happening until it's too late, because all of these invaders each have a house key. And it'd be like giving a burglar your keys to your house, or if writing your PIN code on your phone and then just leaving it out somewhere in public. So these things are just, that's how they get in, and they're not recognized. Then the other type here, like hepatitis B, does exocytosis, and you can probably guess exo means outside, and cytosis means cytoplasm, but out of the cell. So the virus enters through endocytosis, that your cell thinks, hey, come on in, this is nothing to worry about. The cell makes a bunch of copies of it, and then your vacuoles store them, and then they release it just like it was making a bunch of protein, just like it was making a bunch of harmless chemicals or molecules needed by other cells. And your virus has been tricked. Sorry, your cell has been tricked by the virus. Now, there's another stage that we call lysogenic, and most of the time this happens with a virus called bacteriophage. Bacterio means bacteria. Phage comes from a Greek word that means eat, and it literally means bacteria eater. These are viruses that only infect bacteria, and yeah, sometimes we actually use them to fight infections. The phrase, the enemy of my enemy is my friend, that sometimes we can use viruses as treatments to get rid of certain bacteria. But in this diagram that you're going to label in your notes, and you don't have to write it word for word, I want you to just be really straight and to the point about what is happening in each stage. This is just a plain old bacteria. Its DNA is green and it's not yet infected. Next, in stage 2, the bacteriophage attaches and injects its DNA inside. And the red strip here, the red mark, is representing the virus's DNA. Then the DNA goes into the nucleus and becomes part of the bacteria DNA. It is now kind of sneaking in. And then when this bacteria goes through mitosis, goes through binary fission to make another copy of itself, it copies everything, including the virus DNA. And it may do this for a long time, for many, many cycles. It may just make copy after copy after copy of its own DNA plus the virus DNA. And that process repeats, and that's called lysogenic. So as you can imagine, it takes a long time for something to actually happen, and we don't know exactly how long it will take with each virus. Sometimes it happens pretty quickly, sometimes it might not happen for many, many months or years, but we say that until something happens, the virus is dormant, meaning sleeping, and then something will trigger the lytic cycle to begin. And we don't know what always causes that. Sometimes it's an environmental change. There are some that will respond to UV radiation, like when the when the cell gets exposed to UV radiation, sunlight, then suddenly it kind of wakes up the virus, and then the virus separates from the bacteria DNA and starts to do the lytic cycle. Sometimes certain chemicals being introduced. Sometimes the host cell is getting weaker, and maybe it's not enough nutrients, maybe it's under attack by some other virus, and so now the lytic cycle starts, and that's usually when lysis will happen, and now those cells are being destroyed by the virus that just woke up. So here is a side-by-side comparison of the two, and on this part of your notes, I want you to give your opinion. Both of them seem pretty efficient. They both result in what we call exponential growth. It's not just one and then adding one and then adding one and then adding another. Each time it's reproducing, it's making dozens, maybe hundreds or thousands, but they're very different. One of them takes a little longer. This one maybe a little more slowly. They call this one the patient cycle. But in the end, it's releasing a lot. But if you look at this one, it's also releasing a lot, and then each of these go off and do this whole thing again. So your opinion, which one do you think is the most efficient at reproducing and and back up your opinion with an explanation. All right, that's all we have for this note.